An overview of brain metastases. In vivo selection of metastatic brain cells. Genetically modified mouse models. Challenge. Conclusions. References. Dynamic. The metastases of certain brain cancer poses an important challenge for oncologists who seek to eradicate these tumors. The complexity of the blood-brain barrier and aggressive cancers that can penetrate the central nervous system often limit the effectiveness of current therapies. Image peterschreiber.media /. Consequently, many researchers from around the world have turned to various in vitro and in vivo models of brain metastases to better understand how these cancers migrate to the brain, as well as what treatments have a promising future in the clinic. An overview of brain metastases. When a local tumor spreads to different organs, it becomes a systemic disease, which is often much more difficult to control through therapeutic interventions. Unfortunately, several different cancers will often go to the SNC, brain metastases representing up to 80% of all brain tumors. Although lung and breast cancers are the most common causes of brain metastases, other cancers, including melanoma and renal cancer, are also often associated with metastatic dissemination of the SNC. One of the largest targets to treat brain metastases is BBB, because most systemic chemotherapeutic agents cannot penetrate this barrier and reach the tumor. The use of radiotherapy for brain tumors is also associated with certain limits, because this therapeutic approach can irreversibly limit cerebral plasticity and evolve in radiological necrosis, potentially lichen complication, and cranial radiotherapy. Due to the lack of available and effective treatments, cancer patients who have been diagnosed with brain metastases are often confronted with a dark prognosis, which is associated with morbidity, mortality, and cost of treatments, thus leaving palliative care to be the only choice of treatment for many patients with this stage of cancer. In vivo selection of metastatic brain cells. One of the main approaches to the study of brain metastases is the development of specific cell line clones capable of reaching the brain. These cell lines are developed by an in vivo selection strategy, in which the cellular cancer cell line is injected into an intracarotid or intracardiac mouse. All cancer cells that successfully reach the brain of injected mice are then recovered and cultivated. Typical